Hello, I'm here to talk to you today about the relationship between maternal diet, epigenetics, and the development of chronic disease later in life. As we've learned in class, the term epigenetics refers to the processes that induce heritable changes in gene expression without alteration of the genetic sequence itself. Two major mechanisms of epigenetics are DNA methylation and histone modification. These epigenetic processes are critical in determining when and where specific genes are expressed. Alterations in epigenetic regulation of genes can lead to changes in phenotype. Furthermore, studies done on both animals and populations of humans suggest that maternal diet during fetal development influences the way in which a fetus is disposed to metabolic diseases such as obesity later in life. The relationship between maternal diet, epigenetics, and phenotype are nicely illustrated in this study, where pregnant yellow agouti mice were fed a dietary supplement of folic acid, a dietary methyl supplement, resulting in increased methylation at CPG islands of the agouti gene in their offspring, leading towards a darker agouti coat color. The gray bars represent the pups whose mothers were fed a normal diet, while the black bars represent the pups from mothers who were fed the diet supplemented with folic acid. As you can see, the pups whose mothers were fed this supplemented diet developed darker agouti coat colors compared to the control. Not surprisingly, we see this relationship between maternal diet and the epigenetic modification of genes also in humans. The diet of people in Gambia fluctuates depending on the rainy or dry seasons. This study examined the effects of the seasonal diet, measured through biomarkers in the blood, on the methylation status of several genes in the babies of women who conceived either in the dry or rainy seasons. The methylation status of the gene is displayed on the y-axis, and on the x-axis are the genes being looked at. The green bars represent babies conceived in the rainy season, and the brown bars represent babies conceived during the dry season. As you can see, the methylation status of these genes differ quite significantly depending on the nutritional status of the woman at the time of conception. These results show that the maternal nutritional status during early pregnancy causes persistent epigenetic changes at human metastable epithelials. So now that we've seen the long-lasting effects of maternal diet on the offspring working through epigenetic modifications to produce a phenotypic change in the next generation, let's think about what health effects these epigenetic effects due to maternal diet could have on future offspring. The increase in incidence of chronic non-communicable diseases, such as cardiovascular disease, obesity, and type 2 diabetes, over the last 2 decades is likely not solely attributable to genetic and lifestyle factors. Evidence suggests that epigenetic mechanisms allow the developing fetus to adapt to nutritional cues from the mother and adjust its developing metabolic status to produce a phenotype matched to the predicted postnatal environment. Gluckman and Hansen have suggested that the changes induced by maternal under- or overnutrition may reflect an adaptive response of the fetus to environmental cues acting through the process of developmental plasticity to make changes in its metabolism in order to be better adapted to the future environment. Poor maternal nutrition signals to the developing fetus that nutrients are scarce and it will adapt a slower metabolism to conserve energy in response. But if these maternal cues are not an accurate assessment of the environment by the fetus, and the actual environment experienced after birth is different from that relayed in utero, there is an increased risk of developing metabolic diseases such as obesity later in life, because the metabolism set during development is not matched with the postnatal environment. I'd like to close with perhaps the most well-known case study connecting maternal diet and chronic metabolic disease. The Dutch hunger winter was a famine that occurred during an especially harsh winter in 1944, when the Germans blockaded food shipments to the Netherlands in retaliation for Dutch support of the Allies. The study suggested that the effects of the famine on pregnant women had differing effects on the health status of their children later in life, depending on how far along in pregnancy they were when they were exposed to the famine. Importantly, the study showed women who experienced the nutritional constraints from the first trimester of pregnancy did not have reduced birth weights compared to unexposed individuals, but did exhibit increased risk of obesity and cardiovascular disease as adults compared to the controls. Individuals whose mothers were exposed in the later stages of gestation had reduced birth weights and increased incidence of insulin resistance and hypertension. This suggests that the exact timing of the nutritional constraint during pregnancy is also critical in determining the effects of a maternal diet on her offspring. Thanks for listening to my presentation. I hope you enjoyed it.